Good morning. Welcome to our 2021 speaker series. My name is Lale Walsh. I'm the NDF CEO, and today I'm delighted to introduce to you an old friend of the NDF, Dr. Stella Mitrani-Rosenbaum. Most of you know her as Dr. Stella and are very affectionately call her that. She is responsible for discovering the GNE gene responsible for HIBM 20 years ago this year, in 2001. So this is a milestone year for us. And as such, Dr. Stella is also a scientist, obviously, and professor emeritus at Hadassah-Hebrew University Medical Center in Jerusalem, where it is currently 9 o'clock at night. So I definitely don't want to delay this talk, but I will say before we turn it over to Dr. Stella that the Q&A will be managed by our very own Dr. Angela Leck, following her talk. So as the questions occur to you, please put them in the chat box. Stella, welcome to our show. And thank you so much for agreeing to make this presentation. I'm going to turn it over to you to take it from here.
Okay, so thank you very much for this kind introduction. And good morning to you all, or afternoon or evening, wherever you are. It's a pleasure to meet you all again, although it's by Zoom, but it's better than nothing for sure. So today, what I want is to talk about why do we need model systems to develop GNE myopathy treatment. And I want to update you on our cellular models and on the animal models we are developing. And I want to emphasize that these projects are supported by NDF for many years now. So we have this GNE myopathy, and we know the primary cause for the disease. So the question is, what prevents its solution? We all know that the gene and the protein responsible are GNE. You are very familiar with this schema. Protein is an enzyme, and we also know the function of this gene. We know that it's a metabolic enzyme that is responsible for sialic acid synthesis. So as you can see here, we have this GNE, the two functions of GNE, epimerase and kinase, that are functioning in the cytoplasmic part of the cells. Then the sialic acid is synthesized and it enters the nucleus where it is activated. And this activated molecule of sialic acid goes out again and goes to the Golgi apparatus, where it joins the long chains of sugars that are on the proteins or the lipids that then all together go to the membranes. And we can see here the sialic acid is the last sugar on this chain. So we know the factor of the disease, the gene, we know the function. So what it is so difficult to fix it? We should go and right away do it, right? So again, what is the schema for having a therapeutic design? So normally we discover a gene, we know the gene that is responsible for the disease. We manage to know the gene function and what are the mechanisms that are impaired in this disease. And then if we know the mechanism, we can design the therapy. So that is the normal pathway to go from the gene to therapeutic design. So in our case, the obvious option is the metabolic solution. That is, we need to fix the sialic acid levels, assuming that this is what is going wrong, that in the mutations of the gene, sialic acid is not synthesized enough, and therefore we have to supply sialic acid. Okay, the problem is that this has been tried. And as all you know, the trial didn't go very well. Of course, could be many reasons for the trial not to have succeeded, and I will not go into that now. But the thing is whether indeed we do know all the functions of GNE. Maybe the sialic acid is not the only thing going wrong in GNE myopathy, if at all, and maybe we should know other functions of GNE to be sure and to address the whole picture of the disease. So indeed, in many laboratories and also ours, the last many years, people have been looking for novel functions involving GNE, and there have been many approaches to that: cell biology, omics, looking for GNE partners in the cell, looking for the role of GNE in development. And all of those studies have given us a little piece of information, but not enough to determine the real entire picture for GNE myopathy that can explain the problem in the muscle, why the muscle is disappearing. So in fact, we must admit today that the precise mechanism by which mutations in GNE lead to muscular pathogenesis is still unknown. And therefore, if this is again the normal pathway to go from GNE to therapy, going through the knowledge of the function and of the mechanism of the pathophysiology. So for our case, for GNE myopathy, since we are not sure of all the functions and the mechanisms, so we have maybe to skip some of those and go directly from knowing the gene responsible and design the therapy. Okay, so that means that we need a more comprehensive approach to address the problem and not pinpoint specific mechanism or specific pathway in GNE function. And of course, this comprehensive approach is to address the primary effect by gene therapy. I mean, addressing the gene, the GNE gene in our case, to go through gene therapy. So the rationale for this approach is that we can supply a normal GNE to the cells. And in this way, we can mimic a carrier state, because in the cell, we will have both the mutated protein and also the wild type protein. So it will mimic the situation of a carrier individual that is healthy. And what is also important to say is that, in fact, we are pretty sure that small quantities of the normal functioning protein will be enough to overcome the disease mechanism. Why do we think that? Because we know that GNE, the protein, the GNE protein is expressed at very low levels in normal muscles. So we assume that if we can supply even very low levels of the protein, we will be able to go to mimic the carrier state. Okay, so now we know, we want today to know, understand what do we need, scientifically speaking, to implement therapy, any therapy, and of course, GNE myopathy. So first of all, we need the tool to treat, and in our case, the tool to supply normal GNE. And the other factor that we need is a target, so we are able to evaluate the efficiency of this tool. Okay? So about the treatment tool, we know today that AAV vectors are the most efficient and the most safe vectors for gene therapy for the last decade at least. And we know that what we need is to supply, to develop the cassette, the vector that contains what we want to deliver, in our case GNE. We put that, we package that in the AAV vector. The AAV vector will transduce cells, also human cells, of course, because that's what virus knows to do. And I am sure that after the year of COVID, everybody is much more aware of what viruses are doing. So those viruses naturally infect the cells, so they are very efficient in doing so. And then what we want from these viruses is that we'll have to get only a single vector administration. So we will have enough transgene expression to get therapeutic levels, so enough protein to do what we want it to do. And of course, that it will be a long-term treatment, that the efficiency will last for a long time. So indeed, many experiments and many trials have been done for the last several years in cells and also in all kind of animals, small animals, big animals, and of course also in human, and also in different diseases, but specifically in muscle. We are now trials in DMD, lipoprotein lipase deficiency, POM disease, SMA, and many limb girdle muscular dystrophies with trials going on. Specifically, we know that the AAV vectors reach very efficiently muscles through the vasculature. That is when we inject them systemically in the blood. And specifically, we know, because we have tried that ourselves, that AAV8 and AAV-RH74 are viral vectors that allow specifically the expression of human GNE in mice. We also know that this specific vector, AAV-RH74, is being used now in several trials in neuromuscular diseases, in DMD, in animals and in humans, and also in many limb girdle muscular dystrophies. So we think that at least to date, if we had to do the trial today, we would have a treatment tool almost ready. Of course, that we cannot define the exact and the specific and the final tool till we know the efficiency, the real efficiency. And for that, we need a target to evaluate the tool. A target to evaluate the tool can be a cellular model with GNE mutations, where we can see something is going wrong in those cells, and then we can try to fix it by the treatment. And we need also, or we can have also animal models with GNE mutation that can recapitulate, at least in some phases, the symptoms of the disease. Okay, so about the cellular models, what do we have? So till today, patient muscle cultures have been characterized in some laboratories, also in ours, also patient muscle biopsies. So these are the real model, the most real model of the disease, since they are cells from the patients. But all the studies that have been done, also including omics, very comprehensive omics studies, show finally very mild effects of those cells compared to cells from no patient muscles. In many different pathways like signal transduction, remodeling processes, cell survival, and so on. So it was very difficult to use those mild effects as markers for treatment. Okay. So when we look at the animal models that are available, In fact, we can say that there is no animal models for GNE myopathy today. We know that the GNE knockout is lethal in utero, so we don't have any mice to look at. And the knock-in models, where we put in the mouse the mutations or some frequent mutations that we can see in patients, do not recapitulate at all GNE myopathy features. So we cannot treat them because we don't have any problem in those muscles. The present cellular models are not informative at all. And also the animal models are not informative at all. So what we need is to explore systems where the effect of GNE mutation is first of all viable. That means that the organism with this mutation will be alive and will be available for us to look at. And that the effect of those mutations would be more dramatic and not as mild as we have seen till now. And to that aim, what we wanted to do is to develop systems where the knockout of GNE, that is a dramatic change between the wild type, would be viable. Okay? So that is the aim of our project, to develop an animal model or a cellular model where we can see the effect of GNE knockout. Okay, so we have done that also at the cell level in muscle cells and also in vivo in the entire organism. So if we look at the first project we are developing, is to have a muscle cell that will be knockout for the GNE gene. So we have used a primary muscle cell that has been derived from the soleus muscle from mouse. This cell is very viable. It develops and divides very well. And what we wanted to do is to knock out the GNE gene. We have target for this aim exon 3, because we know that if we can excise exon 3, we can get a knockout of the protein. So to this end, we have used 3 different guide RNAs to ensure that we will really get some cleavages in this area. And each of these guide RNAs has been cloned in a plasmid that contains, in addition to the specific guide RNA we have put in here, contains already the Cas9 gene. So the Cas9 protein that we need for this genome editing is already there. And we have also another gene that is the puromycin resistant gene. I will explain in a minute why we have put that. So that when we take this plasmid with the guide RNA, each of them separately or all of them together, and we transfect those Sol8 cells, the plasmid will get into the cells, but not in all of the cells, okay? There is some statistic, not all the cells are infected. But then what we do is we add in the culture this puromycin antibiotic, okay? And what will happen is that the cells will die because puromycin is toxic to the cells, except the ones that have the puromycin resistant gene, okay? The meaning that the plasmid is in those cells. The plasmid meaning has the guide RNA and the Cas9. So we will be able to isolate in this manner the clones of cells that have this, all the factors for CRISPR-Cas, and we can assume that in those cells, exon 3 will be excised, or will be modified. Okay, so what we do when we have those clones, we isolate the clones and we check for each clone, what is the situation, the sequence around the exon 3 that we have targeted with our guide RNAs. So this would be the normal fragment we get if nothing has happened. Okay, it's a fragment of 700 base pairs here. And if we have something less than that, we assume that really something has happened in this region. And this region now is missing something. Okay, so to be sure, of course, we take those clones that are promising and we do a sequence analysis. And indeed, we can see, for example, in this clone 12, we can see there is a deletion in exon 3 of 43 base pairs that results after that on a stop codon and some others after that. So that is certifying that we have a GNE knockout. And here again, another example of a 3 or 5 base pair deletion. Okay, so the genome editing has succeeded. We have different clones with different deletions, and some of them indeed result in stop codons after that, that meaning that the protein cannot be synthesized anymore. So we don't have any protein. And we can then go ahead. We took some of those clones and we did a phenotype characterization, first of all, and then a more comprehensive study with transcriptome and proteome. Okay, so for the phenotypic characterization, we know that normal cells do proliferate. We have here myoblasts. This is Sol8 muscle cells, the normal ones before we did the knockout. They proliferate very well. And then when they are a few days in culture, they begin to do some differentiation. We know that muscle cells differentiate, that means elongate a little more till they can form a fiber, a muscle fiber. In culture, we don't have, in normal culture, in standard conditions culture, we do not get to the muscle fiber completely developed, but we can see the beginning of differentiation of more elongated myotubes. Okay, so now when we looked at our Sol8 GNE knockout, we can see that the cells proliferate very well. So first of all, they are viable. The cells don't die because they don't have GNE. So that's a good thing. And then when we look at the rate of proliferation, we see that on the plate, okay, that the cells, the knockout cells, proliferate a little slower than the wild type. Okay? But eventually they'll get to the 100% confluency in the plate. Okay. Then if we look at the differentiation state of the cells, here we can see wild type Sol8 cells. I don't know if you can appreciate, but we can here see that the cells are a little more elongated and they begin to form a kind of myotube. Okay. And if we look at the Sol8 knockout cells, we can see that the cells are much less elongated. They are still kind of circular. Okay, and you can see here at the higher magnification that they are much more rounded and not so elongated. So it seems that there is a kind of delay in the differentiation state of those cells. Of course, now we are trying to go deeper into that and to characterize other effects of the phenotype. And we have begun to do a transcriptome and a proteomic study comparing those knockout cells to the wild type cells. Okay, so indeed, we hope that by those studies, we will see what functions in the cell are affected by the lack of GNE. So we will learn about GNE functions, first of all. And secondly, we could define new biomarkers on those GNE knockout cells. And those biomarkers could be the target for treatment evaluation in the cell. So we have, if we can define new biomarkers or specific phenotype characteristic of the GNE knockout cells, we can use that to target the treatment by gene therapy or any treatment we can think of. We could have a model to evaluate certain parameters. Okay, so that's for the cellular model we are working on. And now we are also, in parallel, in fact, we are also working in vivo at the entire organism. Okay, so again, first of all, we know that the GNE protein, we have a little problem with that because there is no reliable antibody that can detect the levels of GNE in muscles of human or of mice, probably because the level of GNE is very, very low. So it's very difficult to really follow up the GNE protein levels in vivo in the organism. So before we begin generating a model for GNE myopathy, we thought it could be good to try and solve this first problem. So what we did is to generate or to engineer a kind of tag that would be fused to the GNE protein. Okay, so in fact, we have used what is called a flag protein. It's a small sequence of about 60 amino acids. And this flag protein has very good antibodies that can detect it quite easily. So indeed, we did generate these mice with flag GNE. That means that at the very end of GNE, we have a label, and this label can be targeted by antibodies against flag. We can see that is a very strong enhancing of the liver or kidney GNE. These are the organs where GNE is highly expressed, but even here with the flag, we see it very, very high. But we can also see it quite easily in the muscles of the mice, okay? The quadriceps, gastrocnemius, and other quadriceps. So we can have here a good mice to follow GNE protein. We can look at the expression of GNE protein at different time points in muscle, and also at its localization inside the muscle cell. As for example, what we have looked at is the expression of GNE in the gastrocnemius of those flag mice. And we can see that with age, the expression level of GNE is decreasing. Okay, so even that we start with a low level of GNE, we can see that with age, it goes down and down. So this is a mouse at 8 weeks, so it's 1.5 years more or less, and the level of GNE here is quite low. And we have taken also from those mice, we have isolated muscle primary cells from different muscles. They grow quite well, and looked again in those specific cells at the GNE flag. We can see a strong signal. Okay? And when we look by immunohistochemistry, we can see that in the cells, we have a very strong red flag signal. And if we look at confocal microscopy, we can see that most of the flag is around the nucleus. and also a little higher there. So we are now trying to get a better idea of where exactly there are some organelles where GNE is expressed better. So this flag mouse, GNE flag mouse, allows us to look follow up GNE expression, but mostly it is being used as the platform to generate a knockout muscle conditional mouse. Okay. So what is a muscle conditional mouse? Again, we know that mice carrying the GNE myopathy mutations do not show muscle pathology, and a GNE knockout mice do not survive past the embryonic stage, most likely from brain problems. So in fact, we cannot learn anything on the effect of lack of GNE on those mice. So what we want to do is to generate a mouse which has no GNE, but only in muscle tissue. So we want these mice to be born, to develop, but only in muscle there will be a lack of GNE. So we will be able to follow what are the changes going on the muscle. Okay. So to do that, we have taken advantage of a genome editing system quite old, that's called CRE-LOX, and it works like this. When in a DNA fragment there is a specific sequence, it's a short sequence of 34 base pairs that's called LOXP. These sequences are recognized by our enzyme that is called CRE combinase, and when it recognizes that, it just cut it out and take everything in between the two LOXP sequences out of the genome, okay, out of the DNA region. So if we look at our GNE here, if we had those LOXP small sequences flanking the GNE gene, we would be able, by activating a CRE enzyme, to get them out. So we would get the GNE out of the genome. So that would be a knockout mice. Okay? So what's the difference with the knockout that we know till now? We know it will not be born, so that's not a good thing. So we will use, in fact, we are using a little trick about this CRE enzyme. Okay, CRE, like any other gene, is activated by a promoter, and the CRE promoter is a universal promoter. That means it can work in any cell of the organism. So that's not good for us. We don't want that the CRE would be in all the cells, because then we will have a general knockout and it will not be born. So what we can do is, instead of using this universal promoter, we can use a promoter that is specific to muscle. That is, it will activate the CRE only in the muscle. Okay? So in fact, we will have a mouse with these two LOXP sequences flanking the GNE. We will cross it with a mouse that has a CRE gene under a muscle-specific promoter. So we will get, as the descendants, a mouse that will have both this allele and this allele, okay? So that we will have CRE that is active only in the muscle, and it will recognize the LOXP in the muscle only, in the muscle cells, and will get out the GNE, excite the GNE gene from the mouse, only in muscle. Okay? So it's a good thing. The only problem is that maybe, we don't know, maybe if we have GNE knockout only in muscle, but from the very beginning, when the mouse are in the gestational phase, maybe they will not be born either. So we will not really know what's going on. So we can, in fact, use another little trick with the Cre protein. This Cre protein, instead of using the normal Cre, we can use a modified Cre that is called Cre-RT. And this modification prevents the protein to enter the nucleus of the cells. That is, the nucleus is where the DNA is. So the protein is produced in the cell, but is not getting to the DNA. So it cannot recognize the LOXP and doesn't do anything, in fact. Okay. So what we can have indeed is that only when, so those cells, those mice will be born because they are completely normal, okay? But only when we will inject tamoxifen on the tail of the mouse, the Cre will be allowed to go to the muscle, to the nucleus, sorry, and to recognize the LOXP and to activate its enzymatic function and to get the GNE knockout. That means that we will be able to get mice. The mice will be born completely normal, and at any time we want, when the mice will be adults or newborns or old, we can inject the tamoxifen and get all this system work, meaning that we will have GNE knockout in muscle at a very specific time when we decide to do it. Okay, so indeed what we have done till now is again to target exon 3 in order to incorporate these short LOXP sequences next to exon 3. So for this, we use the CRISPR-Cas again. So we design guide RNAs at these sites. And now it's a little more complicated than with the cells because we don't want only a knockout. We want the guide RNAs to target the site where we want to incorporate a specific sequence. Okay, so there is a step, an additional step here to the normal CRISPR-Cas we have for doing knockouts. Okay, so indeed, that's what we did. So we took our flag GNE mice. Of course, we have to expand the colony to have enough homozygotes, okay, that are manipulated with the 2 guides and the 2 donors at the same time. We did that at the same time to get this mouse, okay, a mouse getting this sequence around exon 3 of GNE. So we had 80 pups that were born, and we screened them for the presence of LOXP2 at this side or LOXP3 at this side. And indeed, we found 6 pups with LOXP2 and 2 pups with LOXP3, but unfortunately, no pup at all that had both the LOXP2 and the LOXP3. Okay, so we need, for the Cre to work, we need that the LOXP will be in the same mouse, okay, the same allele of the same mouse. So this was a little disappointing, but we thought we have to go on. And what we did, in fact, is to look whether of those 8 cell lineages, which of them can transmit this modification to the descendants. That's what we call germline transmitters. And we found that only 3 of these 6 founders could really do that. So we decided that we will now expand this 3 founder colony, okay? So we established a colony that has this LOXP2 as a part an integral part of its genome now. And we will work with these mice now to add the LOXP3 at this site. Okay. So again, we had to expand these mice, and we wanted to have all the odds to our part. So we decided we will do the manipulation to 1,000 GNE flag zygotes. So we added these elements for the CRISPR-Cas, and from 250 pups that were born, we had 45 with LOXP3 at intron 3. Okay, and after making a full sequence to know exactly that everything is there and at the exact right place, from those 45, we have finally 15 candidates that are that have both the LOXP2 and the LOXP3. So these 15 candidates, okay, have to be screened for germline transmitters, and that's the step we are now at. Okay, so for that we have crossed them already with 3 ERT mice with a specific muscle promoter we talked about. Fortunately, this lineage is commercially available, so we have purchased it already and have got it and have expanded and have crossed it with the germline transmitters. But before that, to make sure it's a really muscle-specific lineage, we have looked at the Cre protein expression in these mice to see that indeed it is expressed solely in muscles and not in all the other organs. And indeed, it seems to be the case. So we got ahead and do the crossing. Okay, so the final steps. We believe we are now in the final steps of this mouse. That is, we have those mice. We have crossing them with the Cre ERT to see if those, or some of those at least, are germline transmitters. So we want to get a mouse that has the LOXP sequences and the Cre allele. So then the next step will be to cross this again with the LOXP3 to get homozygous mice with the LOX2 and LOXP3. Okay, and then we will have these mice that should be and are completely normal. So they will be born for sure. And then at different time points, we will inject them with tamoxifen to have the GNE exon 3 excised and get a GNE knockout in the muscle solely. And then, of course, what we will do with those mice is to look at very carefully at the phenotype, at the muscle phenotype, to look for pathogenesis signs, muscle structure, histology, of course, and also look at the muscle performance, the muscle function of those mice. After that, we will do also a more comprehensive study as omics analysis to be able to elucidate the biological and molecular processes that are altered due to the completely lack of GNE. And hopefully, if we really can see some pathogenic processes in the muscle, we will be able to use this model to assess the efficacy of the treatments, of the gene therapy treatment, or in fact, any other treatment we can think of. So we have now a cellular model, the GNE knockout muscle cell that we are characterizing deeply. We are almost already at the last steps of having the conditional muscle GNE knockout mouse. And this, we believe, will be new tools that will allow us a better understanding of GNE functions in muscle first. Then we will be able maybe to establish novel biomarkers in muscle with GNE knockout, and they will be used as targets for therapy assessment. So this is where we are, and I want first of all to acknowledge all our collaborators, the past members of the lab and the present members of the lab that have done all this work. Of course, my long-time collaborator, Zohar Argov, and Rebecca Hafner at the Weizmann Institute that has helped us in injecting all the embryos. And of course, to the NDF for the long support during all these years. And that's it.
Great.
Thank you for your attention.
Thank you, Stella. Thanks for that insightful talk and the summary of where we are with the different models, both cellular and animal models of GNE myopathy. I thought it was a very balanced talk for both scientists and non-scientists in the audience. And hopefully everyone can get something out of that. So please post your questions in the chat or the Q&A. It can be directly related to this talk, or maybe anything at all about GNE myopathy that you're curious about. So I have some questions. But before I start, I think Tara had a question, but I answered that. Lale posted a question. What do you expect to learn about gene functions? And do you think we will see pathology? I'm not quite sure what that's in reference to specifically. Do you have any idea?
Well, I mean, if it's on the models we are presenting, of course, the aim of all the project is to see those those changes, some pathologies, some signs of something going wrong in the muscle. But of course, I cannot guarantee that we will see that.
Yeah.
But since we have tried so many different mouse models, I thought we should try also this one before we say
we close the book. Before we close the book and say it's not possible.
Exactly.
So I'm really glad to hear, the progress you've made with that mouse model, in that you have both now both LOXP sites after many tries.
Yeah.
What is the best case scenario for you in this mouse? So maybe like what age would you like the phenotype to appear? Maybe comment on the severity that is ideal. What would be absolute best result in this model?
The best result, I would say, I mean, I would think that the longer we have a GNE knockout, the best chance we have to see something happening in the muscle. So I think we will want first to inject tamoxifen. That means to activate the GNE knockout system at the very beginning when the mice are born. Then if it works well, we can try also even in utero to do that. But we want to be sure that those mice will be born. So we don't want to miss, so we can do both. But anywhere, if we inject, if we have GNE knockout cells from birth at least, we will, I would assume that we will not see an immediate effect of the GNE knockout on the muscle. I mean, it would be great, of course, for us, but I don't think this would be the case. We assume it will take a little time for the muscle to begin to malfunction or to see something. I really cannot say how, but I would say few months. But of course, we could have finally a good surprise for once, and see that much earlier.
Earlier, yeah. I think the earlier the better, but not too early, but semi-early. Yeah, something that doesn't require aging for up to a year or something, because then it would just slow down all the subsequent studies.
Of course, of course.
But so is it possible that the lack of GNE in muscle can be compensated for by GNE in other tissues, that it can somehow enter the muscle? Is that a possibility even? I don't know.
Well, we know that GNE is made up in the cell. It's not in the serum going around and entering all the organs. But so I guess it will not, but I hope it will not.
I hope it won't. So
I mean, as far as we know from the data we know from GNE, it doesn't seem that it will go out of the cell and get into other cells. It must be into the same cell. So I don't think this would be a problem for us if we don't see anything.
We do, though, have to be prepared for the possibility that even with the conditional GNE knockout, we may never achieve a desired muscle phenotype by knocking out or engineering mutations in GNE, given that we know the sialic acid biosynthesis pathway is a little bit different in mouse than in human.
Right.
And maybe we just can't recapitulate that phenotype. You know, the mouse has that extra CMAH gene that converts the NU5AC to NU5GC, which makes it a little bit different, and the different sugars attached there. So that's definitely something to keep in mind.
That's definitely, of course. But that's why we have also the cellular model.
Yes,
there
correct.
You don't have all those compensations maybe working so strongly. So that could, But of course, we cannot guarantee anything. We are trying all we can think of, but
we're doing the best we can. So Noah Weisleader has a question. He posted, "Thank you for the excellent presentation. Your progress with these models will accelerate work in the field. Can you expand a bit on how you will develop additional biomarkers for the disease with these novel markers?" Maybe he means novel models.
Yeah. Okay. What I meant is, if we look at the cells with the knockout, or the mouse, if we have some pathogenic, some phenotype in the muscle, we will do a comprehensive study on omics, proteomics, transcriptomics, and there we can see differences between the wild type and the model in different pathways, in different genes. And we could then, even if there is something that has nothing to do with, or we don't understand what it is for the muscle pathology, we can see if we can fix that when we treat the cells or the models. So we can define, by the results we will get, we can see what is different, see what is enormously different or very dramatically different, and define that as a marker, a biomarker.
Yeah, on both the transcriptomic and proteomic level. Yeah. And then start maybe screening some patient samples.
Of course.
Some cells or their muscle or serum or things like that. Yeah, because,
depending on what we are looking at.
Yeah, with these slower progressing muscle diseases, it's really important that we have some strong biomarker readouts. Otherwise, it's really hard to tell if a particular therapy has had an effect. And, we're recently finding that out in clinical trials of other diseases, such as, FSHD, where the results are very ambiguous because the biomarker readout wasn't very clear. So this is definitely something really important to keep working on. So I have one more question. I think that's all we have enough time for. So something I'm struggling to get my head around, so you touched upon that GNE may have more function other than just the synthesis of sialic acid. And many others have postulated that too. And it could well be the reason why the ultragenics trial failed. But we don't know because there could be other factors also. If that is the case, that GNE has additional functions, do you think then it's important for GNE to be delivered also into non-muscle tissue when we're considering gene therapy? And to not just to target that to the muscle, but it should go everywhere in case it has additional uncovered functions. Do you know what I mean?
Yeah. I think I know what you mean, but what I think is
should
that
we consider that?
We know from the patients that the only tissue that has a real problem is the muscle, right?
That's true.
So it doesn't matter what function goes around, it is a kind of person-specific function. Because all the other...
Because GNE is expressed really lowly in muscle, in skeletal muscle, compared to even fibroblast or many other tissues. Right. It's one of the lowest expressing in GNE, and it's just, that's, to me, I just can't correlate that in my head. Yeah.
So maybe the function that is altered, sialic acid maybe, or something else in the muscle is critical because there is so little in the muscle. And it's not critical in all the other organs because we have much more. So
right.
even if we have a lower level of function in all of the tissues, it is good enough. But in muscle, we have so little functions that if it's even less than that, so there is a kind of threshold that the muscle cannot really...
That's a good rationale. Yeah, what little there is, it needs to be functional.
Right.
Yeah. Okay. Yeah.
That's what I think.
That makes sense. That makes a lot of sense. Does anyone have any other questions, burning questions, or just let me just check the window. Anything at all about research, about progress in the field? Taras was just congratulating you and saying very fascinating discussion. I know some of this can be quite difficult for the lay audience to follow, but you know, we're always happy to discuss more with the patients. Just, you know, feel free to email Stella or email Lale or me. It's, thank you so much for staying up. I know it's quite late there over in Israel.
Fine. Not so late, it's fine. Thank you.
Yeah, and you know, thank you so much. And I'm excited you'll be getting your third booster shot for the COVID vaccine, and I hope it makes
next week. Yes. So,
okay.
So thank you, everybody, and be safe.
Be safe, everyone. Thank you for listening in. Have a good weekend.